Okay, can somebody confirm you can hear me?
Yeah.
I can hear you.
Thank you. Get to the lab. They're still converting. I'm going to share my screen. Should be able to see my screen now. So what I need to talk about this time in the lab is the unknown project. This is a project that you will work on almost all term long from now on, where you'll be trying to identify an unknown bacterial species. And this is all going to be done online. So it'll be done between you and I. And essentially, I'm going to give you a number, and it's going to represent a bacteria species. And let me show you where you can find that. Did that finish? Nope. Actually, let's get out of that too. The possible species are in the unknowns list, SP20. When I open it, actually, I shouldn't do it this way. Let's download it. So the species are on the left here, and there's about 22 of them. The first right here are gram negatives. The ones down here are gram positives. So I'll give you a number and it represents one of these species. And then you will be performing different tests, which are up here, to try and identify these species. So that's the goal. Any questions about that part? Now, initially, I'm going to pick the number, a number of tests that you will do, and I will start the gelatin hydrolysis test. If everything goes well, I'll actually start it tonight. If it doesn't go well, I'll start it whenever I email you the number. But I'm going to try and send out the numbers tonight. And I will pick the first 3 tests you're going to run. You're going to start the gelatin hydrolysis test maybe tonight. And there's the test results there. I'll explain what this is in a minute. And then on Sunday, if I remember correctly, you'll be doing the gram stain. That'll be this test here. And then on Monday, you'll be doing the PR lactose test. And I'll send you the results to all 3 of these tests on Tuesday. The reason why I'm sending them all together is it's just fewer communications between you and I. And the reason why I'm picking the first 3 tests for you is I went through the logistics of this project and discovered that it will take at least 100 messages between you and I for this one project. And I decided that's just too much for one project. And if I pick the first 3 and send one message, that will be reducing 60 messages between you and I, about 20 students. and I don't remember how many test requests, something like 5 or 6. Oh, I'm wrong if I said 100. It's more like 300 messages, which is just too many for one project. And so I'm going to reduce it by 60 messages between you and I by picking the first 3 tests for you. After that, you will then figure out, let's say you have a gram negative, so that reduces it to these guys here. Enable editing here. So it reduces them by half, and the gelatin, we'll say gelatin negative, reduces it to these guys. And then I'm going to say PR lactose AG. I guess I'll have to explain what AG is. So let's go PR lactose negative, and then that reduces it to these 3. I guess that one has, no, that one's okay. Okay, so that's how you figure out what you might have, and then you figure out a test to identify which species it is among these 3. And this one could be a possibility. So PR mannitol might be a test you'd run. This is unusual then that you can actually get your answer after doing that one test. Most of them will be something else. Another good one would be this isn't a bad one. Nitrate. It narrows it down to 2 possibilities, this one or these 2. Voges proscure could be a possibility. Citrate could be a possibility. And then motility could be a possibility. All right. Oops, that should be there. Any questions about any of this? All right. Let's go back to the project. So the unknown instructions is here. Hopefully you've looked through it. I'm going to discuss it now. So it tells you a little bit about the introduction and the rationale. You can look through that. The project format, you need to isolate the, this isn't the best one. Let me see if I can find a newer one. That's actually the newest date, but that's not right. Oh, well, I'll go with this. This has been changed, and that is you're not going to contain a mixture of 2 bacterial species. You're only going to be given 1 number. You'll then proceed with species identification, running a series of tests to determine the identity of the species. You can use different sources, obviously Berge's lab manual, which you can find copies, or at least partial copies online, your textbook, this lab module or this lab project. You should be using dichotomous logic when you ask for a test. Dichotomous logic is where you involve a series of yes or no or true or false questions. Let me go through Paint and show you that. Which initially we're kind of doing that, and that is we're doing gelatin hydrolysis test first. So we have all species. I'm just going to go all. And then you'll have the gram positive and the gram negative. Let's go gram negative here. Let's blow that up a little. And then we can do the gelatin hydrolysis test, which you're given. And we're going to assume that it's going to be gram positive, so I'll circle that one. We'll circle all. And you have gelatin hydrolysis positive and negative. I'm not going to put hydrolysis in there, but positive. And let's go with gelatin negative. Oops, wrong one. And then we have PR lactose, which for the gram positives, oops, that's not very good. Let's erase that. And you have the PR lactose, which actually I think is going to split into 3. Come on. Sorry, I'm not doing a very good job here. And that is, you can have for PR lactose, you can have AG, A, and then negative. So you're following dichotomous logic. You're doing first one test, and actually you're doing the gelatin first, but I've got it graphed this way. Gram positive, then you're doing the gelatin hydrolysis, and it's gelatin hydrolysis negative. Come on. And then we'll do the PR lactose, which I don't have that up there. So let me put that there, which will go negative. The point is, you're doing one test at a time, and that is following dichotomous logic. Okay, any questions about that? If you ask for more than one test at a time, you will not be following dichotomous logic. There will be a penalty if you turn in your report and you've not followed dichotomous logic. Where are we? Here we are. So follow dichotomous logic. Shotgun logic will be severely penalized. And that's like if you ask for all the tests at one time. That would be an extreme example of shotgun logic. If you asked for lots of tests over lots of days, that would be following shotgun logic. You should record your daily lab notes. Obviously, on a day you're asking for a test, you should put down on that day the test you're requesting. And what you want to do for recording your daily notes is use your unknown notes pages. I won't have an unknown notes because I'm an instructor. I've got a speed grader, but you'll have an upload page instead of a speed grader link. And you click on it and you would get your page uploaded and it'll be right here. Obviously, nobody's submitted anything here, but what you want to do in your unknown notes page initially is put your unknown number in your unknown notes page. And then I'll send you the test results for gelatin hydrolysis, the gram stain, and then the PR lactose. You should put your results, those are your observations. If the tube is yellow, that's your observation. You should then put your conclusion. The test was positive. Okay. So you should have both your results and your conclusions. If it's not clear to me that you're making a distinction between a result and a conclusion, and you're using the terms interchangeably, you will receive a penalty. Although the unknown notes page is only worth 5 points, it is going to count as a lab assignment. Anyways, put everything on your unknown notes page. This should be your daily activities. When you get results that I send to you, you should put the results up there. You don't have to put the pictures, but you may. You may just say the PR lactose tube was yellow. Okay, that's your result. And then the conclusion is the PR lactose was actually, the conclusion is it's A because it's yellow. And then at the end of that document, do I have that there? So at the end of the document, put your test request. If you have your test request at the very top of the page, and every page will be dated when you submit it, or at the bottom, I'll find it. If you have your test request someplace in the middle, I may not find it. Okay, meaning what I'm going to do each day is I'm just going to go through every student, scroll to the bottom, and I'll see the stop when it shows up. So if it's at the top, it's okay. Or if it's at the bottom, it's okay. And then I'll just go to the next student, scroll to the bottom, looking for a test request. And if it's time for me to send the results for the test you requested, I will send it in the comments. Please do not use the comment section. I will use the comment section. If you want to put in a comment, put it in your document that you're submitting. You may upload an unlimited amount of documents for your unknown notes. Okay. You may only have it in one document, like if you wait well, you can't wait till the very end, but let's say you only ask for one test, you could wait until the very end and put in everything in your unknown notes page at the very end. You must have everything for 5 points. Okay. And it states here what you should have in the unknown notes page, your daily activities. You should have your results and your conclusions for a test. You should put in your test requests. You should also record things like, and I don't have that in there, but I put that in there. You should have in here the species under consideration. The unknown notes page can be informal. Oh, there, I got it right there. I just didn't see it. The species under consideration. I was going in there to add that because I didn't see it, but now I found it. All right, any question about your unknown notes page?
Yeah, I have a question.
Okay,
Are you gonna, so are we supposed to submit a new like dichotomy every single time in our notes?
what do you mean?
Like, you were showing us like the, dichotomous,
Dichotomous tree? No, you don't need to do that in your unknown notes page.
okay. And
Okay.
then are we just asking for a random test every time, or are they coming in a certain order?
I told you, I'm giving you the first three
Okay,
tests. After that time, you pick the test
okay.
to
Sounds good.
choose. And it better not be random. You should look on the chart, consider what are your possibilities,
Right.
and then pick a test to identify your species.
Okay, thank you.
Okay. And you do that in your unknown notes pages. Now, the first time, well, actually, the first two times, I'll try and send you your number tonight. And then if I get everything together, meaning I get the number out tonight and I get everything set up for this experiment, I'll send you the results a week from tonight. So that'll be next Tuesday. And the reason why it's going to be Tuesday is the gelatin hydrolysis test takes a week to run. And that's one of the tests I chose because this one takes the longest to run. Okay, so you need to go take a look at the test you request. Let me see if I've got that showing. Let me show that here now. So week 5 lab unknown tests. These are all the possible tests you can run. And you need to read up on the tests you're requesting. And one of the things you can do is find out how long the test takes. Like I told you, the gelatin hydrolysis test takes a week to run. You have to grow it at room temperature and it takes a week to run. Most of the tests take 2 days to run, like citrate takes 2 days to run. You pull that up. Well, this is taking a long time to do. The MR test takes 5 days to run. So if you ask for that test, you're not going to get your results and observations for 5 days. There it goes. Shows you some information about the test, and there's the procedure here. Place the tube and incubate for 2 days. Okay. Most of them do take 2 days. There are a few that are 1 day, like catalase and oxidase. What's that third one? Pigmentation. These take 1 day. Most of them take 2 days, and the MR test takes 5 days. I don't remember any that take longer than 5 days, but are more than 2 days. There might be one in there, but I don't remember. Most of them take 2 days. Now, when you request the test, it'll depend when I look at the unknown notes page. For example, if you request the test late on Friday, and I've already checked the unknown notes pages on Friday. Let's go there. I've already checked the unknown notes page on Friday. then I may not check the unknown notes pages for your test request until Monday, meaning on Sunday, I'm not going to be looking at the unknown notes pages for sure. And on Saturday, I may not look at the unknown notes pages. So during the weekend, I may not look at the unknown notes pages. I will try and look at the unknown notes pages at least every other day, Monday through Friday. So like I said, if you request the test on Friday after I've looked at the test, I may not get it to it until Monday. Usually I look at the notes pages fairly late, like after 10 p.m. So usually I can find them, but I'm just warning you about that. Over the weekend, I may not look at the unknown notes pages. I generally look at the unknown notes pages at least every other day, and oftentimes I look at them every day, Monday through Friday. But on Sunday, I'm not going to look at the unknown notes pages ever. And on Saturday, I may or I may not. Any question about any of that?
Question about the sample report?
I'll talk about that in a little bit.
Okay.
Yeah, but I will get to that. Let me move on to the unknown project. Where the heck is that? Oh, here it is. All right, so your daily notes in your unknown notes pages can be informal. You do not have to be formal. If you do make your unknown notes pages formal, then it'll be very similar to your unknown report. The unknown report is going to be your unknown project typed up and then emailed, or not actually emailed, you put it in the unknown notes project. And let me show you where that is. Oh gosh, I lost my place. It's really close to the unknown notes, and I guess I don't have a place to put it here. Let's scroll down to week 12 or 10, so whatever the last week is. There's your unknown report. So you open that and then you put it in here. You'll have an upload page. I've got the speed grader page, and you just put it in there. The point is that the unknown report can be similar to your unknown notes if you're doing your unknown notes very formal. The two do not have to be similar. You can do the unknown notes pages very informal, but the report should be formal. The report should be written. Please use 12-point font on the reports. I wear glasses. You have less than 12-point font, I'll have difficult time reading it. You should give each day the description of what tests you asked for, what the results were, and usually you'll give the results on the day you give the results. So on day one, you will request the test. Let's say this test took a day to read. On day two, which would be, I don't know, let's say 5, 7, 5, 9, you request a test. 5, 10, you got the results. So put in the results on 5, 10, and each day should be dated. You don't really need the hour in this one, but I think the unknown notes page does put the hour. So discuss what tests you run, the information you hope to gain from the test, the observations you made from those tests, and then the conclusions you made from the test. Do not omit data to make your project look better. Adding drawings, photos, and tables are helpful but not required. I will send pictures. for the results of your unknown notes page, you can add those pictures for your project, your report. You do not need to describe the step-by-step details for each test you run. I know the steps of the gram stain. All you have to do is say, I ran the gram stain on this and this day, and here were the results. The cells were purple. So my conclusion is the cells were gram positive. You should put your thoughts and your words in your report, and they should be your own words. Don't quote from someone unless you put it in quotes and say where you got it from. In your report, but not in your unknown notes pages, you must have either a table or a figure identifying your unknown organism. I've got the next page showing the table. So you're starting with all species, and that's not shown here. And then you have all the gram positives, which are shown. And then it was catalase positive, and that narrowed it down to these 4. And then let's see, what's the next one? It's being hid. PR mannitol narrowed it down to these 3. PR glucose narrowed it down to these 2. And then citrate narrowed it down to Micrococcus varians. Okay. So this table is showing me the steps you used to identify the species. Generally, the tests you show here are the tests you ran, but if you made a mistake or you got the same results, like the next test gave you the same 4 here, you don't need to put that test in the table because it didn't help you identify your unknown. But you should put all the steps you used to identify your unknown in the table for the figure. And the figure can be like a hand drawing. Remember I showed you kind of paint earlier? I think I've already closed it, but I said what's common is people do the gram stain. So we have all. I'm going to just do this very simple here. And then we have the gram positives. And we're going to go gram here. Not too bad. And there's the gram positive, the gram negative. And we'll scratch this. And then you do something to separate the gram negatives. So you can have your drawing something like that. Okay, so it can be a figure like this, or it can be in a table. For most people, if they want to type, use the table, because it's difficult doing it in a figure if you're doing typing. But you must have something like that in the report. You must have a paragraph in your report. It could be as much as 2 paragraphs discussing your microorganism. This will be a description discussing the organism that you think you've identified. You may get the information from Wikipedia. Don't use Wikipedia as a source, but you can get the information and then use it and then just give other sources. So you do need at least 1 paragraph discussing your microorganism you've identified, and you should cite where you got the information to get your information from your organisms. Most students will only have like 1 reference that they're getting for where they get the information about their microorganism, and that's fine. You do need a reference section to state where you got your information from. Do not plagiarize. You could get a 0 on this project if you do plagiarize. At a minimum, you'll get a 5% penalty. The grading criteria is listed below. I'll use the grading sheet. We'll be mainly looking at the logic you use to perform your test, the completeness of the report, the organization, clarity, and neatness of the report, meaning are the lab entries dated, are they following a logical order? All information has to be typed. If you successfully identify your unknown, it'll be worth a small percentage. I don't remember if it's 5 or 10 points. I'll look at that in a minute. It's one of those two. All right, you don't need to work through this procedure. This is something we used in the lab when we gave you a mixed culture that had 2 species in it. So then we grow it on TSA for all the species to grow, azide for the gram positives to grow, and then McConkeys for the gram negatives to grow. And then we streaked it out twice to get a pure culture, meaning we streaked it out for colony isolation 2 times. And then we set up the stock culture. And I'm just going to assume you're given the stock culture to start with. And then you don't need to do the dichotomous procedure, but if you're a little bit confused about the dichotomous procedure, you can work through this. Okay, you don't need to answer those questions either. All right, let me show you how the report will be graded. Scroll back to week 5, lab. All right, the utilization of resources. Was excess media used or irrelevant test performed? If one excess or irrelevant test is performed, you'll lose 8 points. If none, you get the full 10 points. Was dichotomous logic used in selecting your test? If one excess test is used, you'll be down 2 points. If dichotomous logic is used, you'll get 10 points. If you use shotgun logic, you'll use, 8 points. And you have no logic used at all, you'll get 0 points. Were all relevant tests used? If all relevant tests were used, you'll get 10 points. If you ran your test and you omitted one test that I think you should have ran, you'll be down 2 points. 2 or 3 tests omitted, you'll be down 4 points, etc. Were the correct species included at each step? All species are correct, meaning you're identifying what species are possibilities at each step of the process. You'll get 10 points. If there's 1 incorrect species, you'll get 8 points. 2 to 3 incorrect species, you'll get 6 points. I should put on here, because sometimes students don't list any species, in which case, you really should get over 5 incorrect species if you don't have any species listed, in which case you'll lose 8 points. Well, completeness of the report, if everything, all information is presented, you'll get 10 points. If I have 1 instance of missing information, you'll get 8 points. And that could be like, for example, you're missing the references. That would be something that's missing, 8 points. Effective communication, was your English good, your grammar good, the spelling good, the punctuation good? If everything's good, you'll get 15 points. A few errors, but logic is understood, you'll be penalized 2 points. Some errors, but logic is still understood, you'll be penalized 4 points. And if you have some errors and I can't understand your logic, you'll be down 8 points. If the correct species is identified at the end, you'll get 10 points. If you got a species that was similar to what the species was, you'll get 7 points. If you are outside the group of species, but the gram stain is correct, you'll get 5 points. And if you identify a species and your gram stain is incorrect, you get 0 points. So the total project is 75 points. Any question about the grading? All right. Let me see what else I want to talk about with the project, and then we'll talk about what some of these are. Actually, that's it for the project. Any questions about the project? All right, let's get to the sample journal. This is a sample journal of what your journal should look like. Now, obviously, the beginning will be a little different, and that is this instructor, who actually was an instructor, wrote this up way back in 2004, and he streaked out plates to grow up the bacteria, like normally you put it on azide and McConkey's, and that helps you determine if it's gram negative or gram positive. And then you need to streak it out 2 times to get a pure culture. I'm going to give you a pure culture to start with, so you don't really need the beginning here. Although you could give the colony characteristics. Let's move on. Scroll down a little bit. So this looks like he did the gram stain. Perform the gram stain. So he's got observations, activities, and then on most days he has conclusions. On this day, it looks like he put the conclusion in with the he put the conclusion in with the activities. Don't do that. You should put the conclusions in with at least the results or the conclusions part. Here on this day, he's got observations, none. Activities, he's going to start hydrogen peroxide test, which is the catalase test. And conclusion was catalase B was catalase positive. Colony B was catalase positive. So like I said, for each day, like February 9th, 2004, you should have at a minimum your activities, what activities or tests you're asking for, what observations you have on that day, which may be none, and then the conclusions, which also may be none. And he's got it set up observation first, activities, and then conclusions. You don't have to do it that way, but do realize that when you ask for a test on one day, generally speaking, you will not give the observations for that test on that day. So he doesn't have the activity on this day for mannitol broth. He's got the observations, and the activities here he's got he's going to ask for, I think, hemolysis test. Okay. So in each day, you might have activities, observations, and conclusions. And these observations may not be the same as the activity. If the test that you're requesting takes more than 1 day to run, you won't get the activities on the day you're asking. You'll get them like 2 days later, in which case you should put 2 days later the activities, the results and conclusions for the activity you requested earlier. Any question about that? So be time-wise correct. He's showing you the table he used to identify the species. And it looks like he identified this as Staphylococcus aureus. And it mainly came from beta hemolysis. It looks like he's doing two tests to confirm it. You can't do two tests to confirm it, but you can do one confirmation test, meaning if this test narrowed it down to one, you may do one further test to confirm that this is your actual species of identification. Okay. It looks like he did two confirmation tests, and I'm not sure why he did two, because it looks like they excluded the same species. So, all right, any questions about this? So there's the sample journal, there's his references. There's the table he used to identify as unknown. There's the paragraph about his organism. All right. Any questions about this project? All right, if there's no questions, I think we're done. Let me check there. Syllabus. We will talk about this, right? Yes, it is. We'll talk about the Lab 7 on our next lab. All right, that's it for me. Any questions? All right, I'll say goodbye.
I had a question about the infectious diseases project.
Okay.
So for I know you use speed grader, and I mean I know a lot of different institutions use different ones, but what is the limit for the text that is cited? What percentage? If there's a limit of percentage.
You're talking about the plagiarism test?
Yes.
Okay. If it's over 20%, meaning 20% of your paper is plagiarized, I'll look into it. You won't get a penalty unless it's over 30%. Okay.
Look,
Because,
that is
like
directly
I
quoted,
said,
right?
when you have things in quotes and your references will be expected to be plagiarized because you're just putting in the reference. So if you have anything in quotes, obviously that would be it. And I will take that into consideration, but generally less than 20% is good. And you won't get any penalty unless it's over 30% plagiarized.
Mm-hmm.
Okay,
Yeah, that is directly quoted, right?
sorry.
Even with citations, you mean it's
Yeah,
directly quoted.
if you're quoting too much, I told
Mm-hmm.
you I will give you a penalty. I had
Mm-hmm.
one student who turned in a paper where
Yeah.
over 50% of the paper was in quotes. And
Okay. Mm-hmm.
I decided that's too much. Okay. So you won't get a penalty unless it's over 30%. But if the
Mm-hmm.
plagiarism test says that 30% of your paper is plagiarized, I will look into it. And
Mm-hmm.
if you have plagiarism in there, I will penalize you. Okay.
Mm-hmm. Okay. And for the lecture for all the like the citric acid cycle, the Krebs cycle, um, the details, the extent of the knowledge that we're supposed to possess for the ensuing tests, you will let us know, correct? The extent of that.
Are you asking for the lab or the lecture?
Oh, sorry, this is lecture now.
Okay. For the lecture, I will go over that material, and anything
Mhm.
I don't go over, you do not need to know.
Mhm.
Okay?
Okay.
Anything that I do go over, generally you should know.
Mhm. Okay. Sounds good. And the
Okay.
pictures, right? The pictures that will be, you mentioned they're probably going to be included, maybe, right? For
You
the
mean
lab.
for the lab, I'll send you
Sorry
the
for the
results
notes.
for the
Mhm.
notes. In the unknown notes, you do not have to include pictures, but they're generally helpful.
But you will send pictures to
I
us?
will send pictures to you as results, meaning I'm not going to send you information on a test and say the tube was yellow. I'm not going to do that. I'm going to
Mhm.
show you the picture, and then
Mhm.
you're going to have to say the tube was yellow.
Oh, okay.
Okay.
And those pictures will appear in the notes?
You could put the picture itself saying, here are the results, and then you just show the picture. Okay.
Oh, but
You
you will
still
not
have to give
respond in the
the,
notes?
I'm sorry.
You will not respond in the notes, right? You'll respond via email?
Um, Generally speaking, when you use your unknown notes to request a test, I
Mhm.
will send you the results in the comment section of the unknown notes pages.
Mhm.
Okay.
Mhm.
There is one exception, and that is the first document, the first time I send you notes, which I'll just send the notes, not the notes, I'll send an email to everyone, and it'll have the results to everybody's tests, the 3 tests that I'm chosen, and you've got to
Mhm.
look up on the table which is your results with your unknown number.
Mhm.
You got to look that up. But
Right.
that's the only time I'll email you the results. Every
Okay.
other time, it'll be in the unknown notes pages.
Including the pictures, they will be attached, right?
Yeah, I'll send you the results as a picture.
Okay.
Okay.
Mm-hmm. Yeah. Okay. Sounds good.
Okay. Anything else?
Oh, I think it's fine. The only thing is like I want to be no for the I'm trying to be detailed, but maybe I'm saying a little too much. I'm not sure for my labs.
Uh,
Um,
I'll have to look at your labs. I'm not sure
okay.
that I
All
don't
right.
recall. I
All
do
right.
know that you asked 2 questions that I'll try to remember to discuss next time.
Oh, yes. Okay. Yeah.
Okay.
The last lecture.
Yeah, you asked 2 questions. I told you I didn't know the answers. No, I'll try and give those next time. Looking for the 2 questions now.
Mm.
Oh, what did I do with those? Oh, well, I'll have to look for that, but I've got it somewhere.
Okay.
Oh, here it is. Just found it. All right. All right. I will see you next time.
All right. Thank you, Dr. Wolf.
All right. Bye.
Bye.